Taha introduced, there was an interesting program all along. And this last session is going to be even more exciting. I see a paradox in the arrangement of the program, because this is really a beginning and which foreruns all other programs. This is in fact reproduction, and we have a lineup of speakers, eminent speakers, who will take us through right from embryology and then reproduction, the genomics era, and some of the factors that affect reproduction and what would be if something faulty happens in the reproductive process. So with that short brief introduction of the session, let me welcome Dr. Aisha Elaimi from the CGMR and also CIPM to give us the talk on murine embryology and the effect of GM-CSF. Dr. Aisha, please.
Okay, can I start now?
Yeah. Yes.
I would like first to thank the committee of the conference to give me this great chance to talk about one of my projects, which is going to be about the effect of one of the growth factors on the murine embryo's development, aneuploidy, DNA fragmentation, and morphokinetic parameters analyzed using the Embryo Scope. We all know that the IVF culture media is actually a hot topic recently, as many companies started to add active groups like growth factors and cytokines to the culture media. And recently, Aisha set up a group, Culture Media, to look into the composition and the regulation of making culture media for embryo development. It's well known that growth factors have an impact on embryo differentiation, development, and placental morphogenesis, regulation, and energy requirement. So they are actually important for embryo development. Recently, a new culture media was launched by one of the companies containing 2 nanograms of granulocyte macrophage colony stimulating factor based on its potential in promoting embryo development. It's also known as an adjuster of cell development or blastocyst development. It's important for normal fetal placental development and reducing the rate of apoptosis. While some studies have shown that GM-CSF really enhances the development of the blastocyst, other studies have shown that it has no effect. Some studies have shown that GM-CSF has no effect on the rate of aneuploidy or the chromosomal constitutions in human and murine embryos. A randomized controlled trial was performed to examine the effect of the GM-CSF on the rate of embryo implantation. And they actually found that it can improve the embryo implantation and the survival rate, however, only with the female with history of miscarriages. So we aimed in this study to find out, does really adding the GM-CSF to the culture media improve the embryo development? And we did our study where we examined actually the developmental potential of the embryos, the chromosomal constitution, the DNA fragmentation, and we added to the study the embryo development timing analyzed using the Embryo Scope. We know that routinely in the IVF that the embryo selection is made using the morphological assessment using the light microscope. And recently a document was launched that has all the criteria and the terminology of standard for the morphological grading of the embryos at all stages. However, the time-lapse techniques is a technique based on incorporating a camera into the incubator. So it allows the possibility of continuous morphological observation of the embryos based on images that taken for each individual embryo, together with ensuring the stability of culturing condition by avoiding taking the embryos outside the incubator. It also provides a detailed markers of the embryo development and the cell division. And actually, many studies and publications have really found that there is a relation with the embryo development and the cell division, especially the first stages of the embryo development, from the time of fertilization up to the 5-cell stage. These are some of the publications that actually had been done on human and non-human samples, and they found really novel markers that correlate the timing of the embryo development together with its potential to develop to the blastocyst stage. So what we did in our study, we did a superovulation of the female mice. After that, we did a mating for female mice with male mice for the fertilization to happen. After that, the female mice were sacrificed and the 2-cell stage embryos were recovered from the oviduct. These embryos were randomly divided into 5 different groups to be cultured in 5 different concentrations of the GM-CSF. These 5 different concentrations were selected based after reviewing relevant studies on the effect of GM-CSF on different species, together with a control group where no GM-CSF added. These embryos were cultured for 3 days in the Embryo Scope until they reached to the blastocyst stage. The monitoring of the development of these embryos was happened or performed using the Embryo Scope based on the images that were taken for each and every single embryos. After that, on day 5, these embryos were divided into 2 groups where they analyzed either for aneuploidy or for DNA fragmentations. So, as in our study, we had in vivo fertilization, so it wasn't possible to predict precisely the time of fertilization. So we started our time point from the first cell division that happened. And we calculated the duration of the cell division, so the time the embryos that take to transfer from 2-cell stage to 3-cell stage, and from 3 to 4 and from 4 to 5, rather than taking the specific time point. For example, the embryo at that point became a 2-cell or 3-cell or 4-cell stage. For the aneuploidy screening, we use the FISH technique, and for the DNA fragmentation, we use the tunnel test, which enable the assessment and the quantifications of the nuclei or cells with DNA fragmentation. This is an example for 2 embryos that were monitored using the Embryo Scope. We can see that we will see at the end of the video that both embryos actually managed to reach the blastocyst stage. So morphologically, we can say that both embryos have a high potential to develop to the blastocyst stage. But if we went back to the start of the movie, you can see that this embryo transferred to the 4-cell stage before that embryo. So the second embryo was slower in its division. And the more time the embryo takes to transfer to the 4-cell stage and the 5-cell stage, the more that exposes the embryos to have any errors, either for chromosomal or DNA fragmentation. These 2 images below, they just show an example in how we analyze the DNA fragmentation. The first image, the cells stained with the DABI to show the number of cells that we have after we fix them on the slide. And those 3 cells that stained with the fluorescent green, those are the cells with the DNA fragmentations, meaning that 3 cells that did not show here has no DNA fragmentation. So, in the result, when we compared our control group to the GM-CSF group, we found that the rate of blastocyst formation actually found to be higher in the control group where no GM-CSF added. And by the time that every time the concentration of GM-CSF increased, the rate of the development decreased. And this could be explained by the negative regulation of the GM-CSF receptor when it presents at high concentrations. When it comes to the aneuploidy rate, we looked at the data from different angles. So first, we have calculated the number of aneuploid or mosaic embryo, and we found that the number was lower in the control group comparing to the GM-CSF groups, although the difference wasn't that significant. And then we looked at the range of the aneuploidy. For example, in the control group, the range was from 0 to 15 or to 10. In the GM-CSF group range from 10 to 30. So we calculated that range and we found it lower again in the control group comparing to the GM-CSF group. The last things we did, we looked at the number of the nuclei or mosaic nuclei in each and every single embryos. So for example, if I have a blastocyst that with 100 or 150 cells, how many of these cells were aneuploid cells? So we calculated the proportion of aneuploid cells in each embryo, and then we calculated the group, and that was again lower at the control group compared to the GM-CSF groups, although it was non-significant. When it comes to the DNA fragmentation, again we followed the same way to look at the rate of DNA fragmentation. So the number of blastocysts with DNA fragmentation, there was no difference between the control group and the GM-CSF group. And the average rate of DNA fragmentation, again, no significant difference. But when it comes to the proportion of the cells or nuclei with DNA fragmentation, it found to be significantly high, significantly different in the 2 nanogram group and the 5 nanogram group. And this could be explained by that the GM-CSF induced the synthesis of anti-PCL2 families in the human neutrophil cells, which therefore reduced the rate of killing the cells with DNA fragmentation. So it reduced the rate of apoptosis by reducing the synthesis of anti-apoptotic PCL2 families. So it allows, in another word, it allows their cells with DNA fragmentation to survive. In another word. The most important point is the time lapse that we looked for. We wanted to know precisely, does really the GM-CSF affect the development? Does it enhance the cell rate, reduce? Does it make an effect? We actually found no significant difference. We calculated the time the embryos that take to transfer to the 3-cell stage. None of the GM-CSF group showed significant difference comparing to the control, to the 4-cell stage. However, you can see in some of the group, the 2 nanogram group comparing to the control, it takes actually longer time to transfer to the 4-cell stage comparing to the control group. Even the 5-cell, the T5 as well was longer in the GM-CSF group comparing to the control groups. So to conclude, we found that our study showed actually a decrease in the blastocyst development in all the 4 GM-CSF concentration compared to the control. No significant difference found in the aneuploidy or mosaicism or the ratio of the aneuploid between the control or the GM-CSF groups. However, maybe the high number of aneuploid cells in the GM-CSF group could be explained by that. We know that in the cell there is a mechanism that called checkpoints that responsible for holding the occur at the metaphasis stage. It's responsible for initiating the signal that we call it hold, where all the chromosomes align, so can the spindle bind to all the chromosomes before the transition to the next stage, the anaphase, where the 2 chromatid, the sister chromatid, separated. So this action that happened in the cell regulated by interaction of several proteins. And they found that one of the genes called MAD2 genes in the mice is really affected by one of the receptors of BCL2 family, which is part of the GM-CSF family. So maybe the disturbance in the GM-CSF in the culture media affected the function of that gene, which led to the mis-segregation during the development, which increased the rate of aneuploidy. We saw a significant difference in the mean cell number with the DNA fragmentation between the control and some of the GM-CSF group. And finally, no significant difference as we saw in the cleavage time events with the addition of the GM-CSF compared to the control. So adding the GM-CSF to the cultural media actually affected negatively, we can say to some extent, the embryo development. And more studies on larger numbers on the embryos and more randomized control trial actually required to evaluate the effect of GM-CSF on embryo development, rate of pregnancy, and most importantly, the live birth. And embryos and the baby after birth, actually, most important. And thank you. And I would like at the end to thank my PhD team at the University College London, especially my supervisor, Joyce Harper. And I would like to thank also the group at the CGMR and King Abdulaziz University for funding this project, and Unisense company for providing us with the Embryo Scope to perform this study.
Interesting research. Thank you, Dr. Aisha. The floor is open for questions. Okay, let's move on to the next presentation.